So students, after the basic biomedical instrumentation we studied in the last class, the next heading we are going is for the origin of biopotentials. As I told, biopotentials are nothing but the voltages that originate from the human body. Okay, biopotentials are usually ionic voltages. What are ionic voltages? The voltages that occur due to the difference in the concentration gradients. That is what is called as the ionic voltages, and they are in the range of millivolts. They have to be amplified, produced as a result of electrochemical activity of the cells. So electrochemical activity of the cells in the sense, some positively charged and negatively charged ions are there surrounding the electrode and in the electrolyte. So what happens is, these ions are responsible for the charges inside and outside the cell. That is what we are going to study elaborately in the upcoming session, in the current session. So these are nothing but the ionic voltages. These ionic voltages are to be converted to electrical voltages, measurable electrical voltages. The natural signals can be measured and displayed to aid the physician to diagnose various diseases. So these electrical voltages are useful for identifying the diseases. That's what we saw. That's what we are going to see in ECG, how they are going to diagnose. So that is nothing but the amplified ionic voltages that are called as the electrical voltages. So ECG as electrical voltage is finally useful to identify the normality and abnormality of the heart. That is what the origin of biopotentials, introduction. And then what is meant by resting potential and action potential? So before going into the introduction for the resting potential and action potential itself, we will see the final outcome, final graph, what is meant by resting potential and action potential itself. That helps in some situations. When you see the final graph and then go to the understanding of the basic concepts, that may be helpful. So resting potential is something where the cell is negatively charged. You may be able to infer the value here, minus 70. So minus 70 millivolts is a voltage that is inside the cell membrane. That is called as a resting potential. And when the cell is excited, so this is a resting state. When the cell is excited, what happens? The cell becomes active. The cell goes to activeness. That is also called as action potential. This process of transition from resting state to action potential, that process is called as a depolarization. So you have to remember, it is depolarization. It may be confusing. In the initial setup itself, it is a negative phrase is coming here, but actually when the cell is in the resting state, it is said to be polarized. When the cell is in the resting state, it is said to be polarized, where the inside of the cell is negative. It is called as a resting potential or polarized condition. From resting potential to action potential, that is called as the depolarization, and now the cell will be said to be depolarized. And after that, what happens? The cell again comes back to resting state. Then that process is called as repolarization. And now this is called as a resting state or polarized state again, not a repolarized state and all. Polarized state or depolarized state, again polarized. But the process, this is called as a depolarization, repolarization. Repolarization in the sense, it again goes to polarization. That is called as the repolarization. With this, so what is a resting potential and what is an action potential? So before going into the complete theory, let us see what is a resting potential and action potential. The final outcome in a graph. What happens in a resting potential and action potential? Suppose the cell is in a resting state. What happens is, when it is in the resting state, the cell inside is more negative and outside is more positive. So when there is a change, when something excites the cell, so we can understand this first and we can go into the details later. When the cell is in the resting state, the cell is negative inside. So this is called as a resting state or resting potential. So then what happens? Some disturbance is there to the cell. Someone touches or some excitation, some form of excitation is there, means the cell comes from resting potential to action potential. There what happens is, the inside of the cell was previously negative. Now the inside of the cell becomes positive. So this is minus 70. I think you are able to read. In the resting state, inside of the cell is negative. When it is excited, it goes to positive. The inside, we are talking about the inside of the cell. That is very important. You take into consideration thinking both the things, it will be confusing. So think about the inside of the cell. Inside of the cell is negative. That is called the resting state. The cell is very resting. So the inside of the cell is negative. So when some excitation is there, it goes to the action potential, where the inside becomes positive. More importantly, this process where the resting potential is taken to action potential, that process is called as a depolarization. Very importantly to remember, depolarization. First itself it comes with the negative term. Why? Because It comes from resting potential to action potential. When the cell is in the resting state, that is called as the polarized state. When the cell is in the resting state, that is called as the polarized state. When it goes to action potential, that is called as the depolarization. And after some time, again from action potential, it comes back to the resting potential. That is called as the repolarization. Polarized state, depolarization, and repolarization. So when I talk about repolarization, I am moving here. That means this process is called as the depolarization. This process is called as the repolarization. So depolarization, repolarization are all processes. Whereas the state, this is called as the resting state, this is called as the action state or action potential. So we need not restrict with this. We have to study in depth why this resting potential becomes action potential. First of all, what is resting potential by itself? What is by resting potential? We will see. Cells are encased in a semi-permeable membrane and permit only some substances to pass through the membrane while others are restricted outside. Rather than going into very elaborate discussion, I can tell you very clearly, sodium ions are blocked outside the cell, whereas potassium ions and chloride ions, that's why I have very clearly given it in color. Sodium ions are blocked outside the cell, whereas potassium ions and chloride ions are free to move inside and outside the cell. So sodium is positively charged, potassium is also positively charged, and chloride is negatively charged. The positively charged sodium ions are blocked outside the cell itself. So what happens automatically, as more and more sodium ions accumulate outside the cell, the outside of the cell becomes more positive. So automatically the inside of the cell becomes more negative. That is what is called as the resting state of the cell. Surrounding the cell body, there are body fluids. These body fluids are called as the electrolytes. These fluids contain the charged atoms like sodium Na+, potassium K+, and chloride C-. The cell membrane readily permits the entry of potassium and chloride ions, whereas it effectively blocks the sodium ions. The ions seek a balance in concentration and electrical charge between inside and outside of the cell. So naturally a balance will be sought. So natural tendency is not to allow a negative charge or positive charge. They want to become neutral. The inability of the sodium ions to penetrate results in two conditions. What are the two conditions means? The first condition is the outside of the cell becomes more positive. That is what is the first condition. The concentration of sodium ions inside becomes much lower. Since the sodium ions are positive, this would tend to make the outside of the cell more negative, more positive, or inside of the cell more negative. And what is the secondary point? The secondary point to remember is, anyhow sodium ions are blocked outside, but potassium ions that are also outside and positive, they tend to move inside, and some of the potassium ions are also allowed to enter inside. So that is the secondary point. The primary point is sodium is blocked outside, which is positive, so inside of the cell is negative. The secondary point is potassium ions are not that much blocked, so potassium ions enter inside. So within the inside of the cell, what are there? More chloride ions are there, some potassium ions are there. So the inside of the cell is more negative. So secondary, additional potassium ions also try to enter inside, causing a high concentration of potassium ions inside. However, charge balance cannot be achieved because more negative ions, negatively charged chloride ions are there inside. Negatively charged chloride ions are there inside. So charge balance cannot be achieved. So even though charge balance cannot be achieved, this is called as equilibrium. So temporarily we call this as the equilibrium. Equilibrium is reached with a potential difference across the membrane, negative on inside and positive on outside. Now we measure the potential here. If we measure the potential here, the inside of the cell is somewhat measuring about minus 70 millivolts. This is called as the resting potential, and that is measured within the cell, which is the negative potential. This membrane potential is called as the resting potential, and it is maintained until some kind of disturbance accepts. This membrane potential is called as the resting potential of the cell, and it is maintained until some kind of disturbance upsets the equilibrium. Some external disturbance is there, means it will change from resting state to action state. A cell is said to be in resting state, it is said to be polarized. The cell in the resting state is said to be polarized. The resting potential of the cell is given as negative, reported as minus 60 to minus 100 millivolts. So at the resting state it is negative. When a section of the cell membrane is excited. Now coming to the next one. So it cannot be in the resting state for the whole time. It has to be. So what is the resting state? Inside of the cell is negative. More sodium ions are outside. more chloride ions are there inside. So chloride ions being negative, the inside of the cell is more negative. So when it is in this state, it is called as a resting state. The other name is called as a polarized. That is what is given in the last line. And after that, the cell cannot be in a resting state for a long time. When a section of the cell membrane is excited, that's what I told simply as some external disturbance. So what happens when the external disturbance takes over? Some ionic current or by some form of externally applied energy, the membrane changes its characteristics. The cell membrane previously does not allow the sodium ions inside, so it suddenly changes its characteristics and begins to allow some of the sodium ions to enter. Some sodium ions are allowed to enter. The sodium ion movement inside constitutes more ionic current across the membrane. So because of the ionic current, the cell membrane becomes permissible to sodium ions. So because of the sodium ion movement, now the conductivity to the sodium ion further increases. This is called as the avalanche effect. That further reduces the barrier of the membrane to the sodium ions. So there is a net avalanche effect. So because of the ionic current, more permeability. Because of more permeability, more ionic current. In which sodium ions literally rush into the cell. More and more sodium ions rush into the cell to reach a balance with the ions outside. At the same time, potassium ions, which were already higher in concentration inside the cell during the resting state, try to leave the cell but are unable to move as rapidly as the sodium ions. So previously the secondary point we studied in the resting state, this is a secondary point for the active state, action potential. So what happens is, here every time the potassium ions are the secondary point. So potassium ion try to leave outside the cell, but they are not able to move as fastly as the sodium ions. So what happens? There is more and more positive charge inside the cell due to the imbalance of the potassium ions. This potential is known as the action potential and it is approximately around plus 20 millivolts. At this condition, the cell is said to be depolarized. So depolarized means again condition. Depolarization means it is a process. So now it is depolarized. So when you look at the graph, you can go to the graph. So this process is called as depolarization. When you end it as ON, depolarization, it is a process. But the state you call it as the action potential or depolarized state. Similarly here, polarized state. Okay. So approximately plus 20 millivolts in the action potential or polarized state. Sorry, depolarized state. Figure 2 shows the ionic movements associated with the depolarization. Previously we saw the ionic motions connected with the resting state. Now the ionic movements connected with the action potential or depolarization. What happens? More and more sodium ions are entering inside. Very few potassium ions are leaving outside. So what happens as a net result? More positive charges are there inside. Once the rush of the sodium through the cell membrane has stopped, the ionic currents that lowered the barrier to sodium ions are no longer present. Again coming back to the resting state. What happens is the ionic current is not there. The semi-permeability of the cell membrane regains. What is that semi-permeability? Blocking the sodium ions. So the membrane reverts back to the original selective permeable condition. By an active process called a sodium pump, the sodium ions are quickly transported to the outside of the cell. So previously in the depolarization, what happened? Coming to the action potential state, more and more sodium ions entered inside. So again these sodium ions are pumped outside. Why means again the cell becomes semi-permeable to sodium ions. So sodium ions are not allowed. So sodium ions are quickly transported to the outside of the cell and the cell again becomes polarized. So polarized means resting state. This process is called as the repolarization. Previously depolarization, the rising part of the graph is called as the depolarization and the falling part of the graph is called as the repolarization. Again one important biological terminology, this is called as a sodium potassium pump. Again letting go the sodium ions outside, that is called as a sodium potassium pump. So this shows the typical action potential waveform of the resting and the action state. So in the resting state it is minus 70 millivolts, in the action state it is plus 20 millivolts.